Welcome back, everybody, for another deep dive.
Yeah,
You ready to get into it?
absolutely.
Awesome. Today, we're diving deep
Ooh,
into the world of vision.
love it.
We're talking photoreceptors,
Yeah.
rods,
Right.
cones,
Yes.
and all the amazing things that
And they were.
make it so we can see the world.
So cool.
And you're not going to believe this.
Okay.
Our sources for this deep dive
Yeah.
are a YouTube video transcript
Okay.
about cyclic AMP signaling
All right.
and a whole chapter
Whoa.
from a book
Mm-hmm.
about thyroid hormone receptors.
What?
Crazy, right?
That sounds wild.
But trust me on this.
Okay.
These seemingly totally unrelated
Yeah.
topics
All right.
have some really cool insights
I'm intrigued.
into how our eyes actually work.
All right.
So you ready to have your mind blown?
Let's do it.
Let's do it.
All right. So sometimes the most profound understanding
Yeah.
comes from some of the most
For sure.
unexpected connections. So let's start with this puzzle
Okay.
from the world of cardiology.
Cardiology.
Yeah, the heart.
Okay, I'm listening.
And this puzzle comes from that YouTube video transcript.
Sure, lay it on me.
So, cardiomyocytes.
Cardiomyocytes.
Yeah. Those are the muscle cells
Okay.
that make your heart beat.
Gotcha.
They have two different
Okay.
types of receptors.
Two different types.
Yes.
Okay.
But, get this,
What?
they both activate the same signaling pathway
Okay.
that involves cyclic AMP.
Interesting. But I feel like
Uh-huh.
there's going to be a twist here.
Oh, you know there is. So here's the kicker.
Okay.
Despite the fact that they activate the same pathway, stimulating these two receptors has totally opposite effects on the heart.
Okay, hold on. So you're telling me the same pathway
Yeah.
can have opposite effects.
Completely.
How does that even work?
I know, right?
Huh.
So that's where this idea of microdomains comes into play.
Macrodomains, okay.
Yeah.
What are
So
those?
Imagine like tiny specialized compartments within the cell, and each of those little compartments has its own set of signaling molecules.
Okay.
It's like having these separate little workstations
I like that.
in a factory. That
Make sure it's like you can have a dedicated space for like baking a cake and a separate space for like, I don't know, building a bookshelf.
Yeah, exactly.
All in the same house. You don't want like flour flying around when you're hammering nails.
Exactly. You want to keep those processes separate.
You got to keep them separated. So in those cardiomyocytes,
Yes.
these microdomains,
They help explain why you can stimulate different receptors, activate the same pathway, but get different results.
because it all comes down to what's in each of those little compartments.
Exactly. It's all about the location. Location.
Location.
Yeah.
Okay, I'm with you. So these microdomains
Yeah.
keep things organized,
Totally.
but what creates them?
Right. That's the question. So there's this group of proteins
Okay.
called A kinase anchoring proteins.
That's a mouthful. Can we shorten that?
Yeah, we can call them A.
AKPs.
Okay.
Much better. So these AKPs.
So the architects of these little compartments, they act as scaffolds,
Okay.
anchoring this key enzyme called protein kinase A. We'll call it PKSI.
PKA. Okay, so the AKPs hold on to the PKA,
Precisely.
and PKA is involved
Yeah,
in all sorts of different
billions of signaling pathways.
signaling pathways.
Even in your eyes.
Whoa, okay.
Yeah, and get this. There are over 50 types of AKPs in humans.
That's a lot of organization.
Right, like we were saying, super organized.
Super organized. So each type of AKA binds to?
It binds to a specific form of PKA.
Okay.
It's like making sure the right signals get to the right places
At the right time.
at the right time.
Okay, so the AKPs are like the organizers. So
True.
they're making sure the PKA is in the right spot to do its job.
Exactly.
That's really cool.
But how do they hold on to PKA?
Yeah. How do they actually physically hold on to it?
Right. It has to be
Yeah.
a pretty specific interaction.
It got to be like a perfect fit or something.
Yeah. So this is where we get into the world of molecular structure.
Let's do it.
So AK cups
Yeah.
have this special structure called an amphipathic helix, and that's crucial.
Amphipathic helix.
Oh, good.
Okay, that sounds familiar.
Yeah.
Like from chemistry class.
That's back in the day. But to remind everyone,
Yes. Remind me.
Yeah.
What does amphipathic even mean?
so an amphipathic molecule
Yeah.
has two sides.
Two sides.
Yes. One side is attracted to water.
Okay.
We call that hydrophilic.
Hydrophilic, water loving.
Water loving.
I remember that.
And the other side
Okay.
repels water.
Okay.
That's hydrophobic.
Hydrophobic, water fearing.
Exactly.
Oh, okay.
So it's kind of like a magnet
Like that.
with its north and south poles.
Okay, so how does this two-sided thing help the AK piece bind to PKA?
All right, so this unique amphipathic helix fits perfectly into a specific binding site on PKA.
Ooh, so it's like a key in a lock.
Exactly like a key in a lock.
Okay.
The hydrophobic part of the
Okay.
helix snuggles into this hydrophobic pocket on PKA.
Gotcha. And then the hydrophilic part
Right.
interacts with all the water
Exactly.
that's around. So it's that perfect fit.
Yeah. Thanks to the amphipathic helix that lets those AKPs anchor the PKA.
And create the microdomain.
And create those microdomains that we were talking about.
It's amazing how these tiny little structures
I know, right? It's mind-blowing.
can play such a big role in organizing ourselves.
And this is happening in every cell in your body.
Whoa.
Including those photoreceptor cells in your eye.
Okay, wait. So we've talked about
Yeah. We talked about microdomains.
the microdomains
Yes.
and the AKPs
Yes.
in the heart.
But how does this relate to vision?
Right. That's a great question. So that's where we bring in our second source,
Okay.
the chapter on thyroid hormone receptor.
Thyroid hormone receptors.
Yeah,
Okay, this might seem like a leap.
like a total leap. What does thyroid hormone have to do with vision?
Well, thyroid hormone
Is
essential for a lot of things.
Okay.
It's essential for various aspects of development, including a healthy visual system.
okay. So you're saying that a hormone
Right.
that's known for regulating metabolism
Right. It plays a role in how our eyes develop.
also plays a role in our eyes.
Yeah.
That's crazy.
Isn't that wild? And this chapter specifically highlights
Okay.
the importance of the TR2 receptor.
TR2 receptor. What is that?
That's a type of thyroid hormone receptor.
Okay.
And it's super important for the development
Okay.
of green cone photoreceptors
Green cone photoreceptors. What?
in the retina.
So it's highlighting the fact that vision isn't just about our eyes.
Exactly.
It's about hormones.
It's influenced by hormones
Yeah.
and signaling pathways.
Throughout our whole body.
Yeah.
Wow, this is blowing my mind. I'm starting to see
Okay.
how this is all connected. We've got the microdomains
Created by ACAPs.
created by the ACAPs,
Right.
keeping those signaling pathways organized.
Right.
And now we've got hormones, like thyroid hormones,
That can
influencing the development
Mhm.
of those photoreceptors.
Right. But how does this all...
But how does this all come together to explain
Yeah.
how we actually see?
So remember those photoreceptors?
Yes,
Yeah.
the rods and cones.
The stars of the show. They are the key.
Okay. And just to recap,
Mhm.
rods are for
Rods are for dim light.
dim light.
Cones are for
Cones are all about color vision.
color vision.
Yes.
But how do they actually work?
All right, so that is where
Yeah.
things get really exciting.
I'm ready. Hit me with it.
We are about to enter
Okay.
the world of the phototransduction cascade.
The phototransduction cascade,
Yeah.
it sounds almost magical.
It is a remarkable process. And to understand it,
Okay. Tell me more.
we need to delve into
Okay.
the
Got it.
intricate molecular mechanisms that are happening
Okay.
inside those rods and cones. I'm ready. I'm following you. Lead the way.
But before we do that,
Okay.
let's take a little detour.
Ooh, a detour.
Yeah.
I like detour.
To another part of the nervous system.
Okay. I'm listening.
That might seem a little unrelated. At
Okay,
first.
hit me with it. Let's talk about the spinal cord.
The spinal cord.
Yeah,
Okay, this is a hard left turn, but I'm here for it.
trust me. It'll all make sense in the end.
No, I trust you.
Okay, great. So within the spinal cord,
Okay.
there's this neurotransmitter called glycine.
Glycine the spinal cord. I'm so lost right now,
Yeah.
but I'm also very intrigued.
It'll all come together.
Okay.
I promise.
I have faith in you.
Okay, good. So glycine is super important in the spinal cord.
Okay.
It's actually the primary inhibitory neurotransmitter.
Okay. Inhibitor. So it's like the brakes.
It's like the brakes of the nervous system.
Nervous system.
Yeah. It prevents things from getting too
Precisely.
far.
And you wouldn't want our muscles
Yeah,
to be
yeah.
constantly firing. Right. We need to control that.
We need those brakes.
Yes, we do. So in the spinal cord, you've got these motor neurons.
Motor neurons.
Yeah. And they control your muscles.
Okay, so those are the ones telling our muscles to
Oh,
move.
exactly.
Okay.
And these motor neurons, they get input.
You receive information.
Yeah, they get information from all kinds of different places,
Okay.
including these inhibitory interneurons.
Inhibitory interneurons. Those sound important.
They are very important. And these interneurons, they release glycine.
So they're like telling the motor neurons
You're like, hey,
to chill out.
calm down. Don't get too excited.
I'm going to be sending constant signals to my muscles.
Exactly.
Okay. So it's all about
balance.
balance.
Yeah.
Gotcha.
We need those breaks.
We do need those breaks.
And this communication, this fine-tuned control,
Yeah.
it happens at the synapse.
The synapse.
Yeah.
Remind me what that is again?
Oh, that's that tiny gap
Oh, right.
between neurons.
Okay. It's like
it's like the
little bridge.
Oh, the little bridge, yeah.
Between them.
So when glycine is released into the synapse,
Okay.
it binds to glycine receptors.
There are glycine receptors.
Yeah, the motor neuron. So it's like
Okay, so what happens when it binds?
when glycine binds to its receptor.
Yeah. What happens?
It causes the receptor to open up.
Like a little door.
Yeah, like a little channel.
Okay.
And that allows chloride ions to flow into the motor neuron.
Okay.
And that influx of negative charge.
Okay, that makes sense. Because chloride's negative.
Exactly. It makes it harder for that motor neuron to fire.
To send a signal.
Yeah, to fire.
An action potential.
An action potential. That's the signal.
That's the electrical signal
Okay.
that tells the muscle to contract.
Gotcha.
So it's not like glycine completely shuts down.
It's
like
turning down
the volume.
Yes.
Like adjusting the sensitivity.
Okay. It's like dimming the lights. Exactly like dimming a light switch.
Instead of
turning them off.
Instead of flipping it off completely.
Yeah.
And this allows for
so much control.
Really precise control over muscle activity.
That's so cool.
Isn't that neat? And so these glycine receptors.
Yeah. They're just like, let's just like.
Those receptors.
In the cardiomyocytes.
In the cardiomyocytes.
Yeah.
They can exist in different states.
Different states? What do you mean? So even though.
Yeah. So even though.
The same neurotransmitter is binding.
Mm-hmm, the receptor can react differently.
Hmm.
Depending on its state.
That's wild.
Right. So they can be closed and resting.
Okay. Like it's just chilling?
Yeah, just hanging out.
Okay.
Or they can be open.
Okay. The channel's open?
The channel's open.
Yeah.
Or they can be closed but desensitized.
Closed and desensitized.
Yeah.
So it's like, it's there.
It's there.
But it's not listening.
But it's not listening.
Okay.
So it's like
another layer.
Another layer. Of control.
Of control, yeah. This ability to switch between states is like...
It's like fine-tuning.
It's fine-tuning the signal.
Yeah.
And this applies to the receptors in your eyes, too.
Well, okay, hold on.
Yeah. They can also exist.
Our eye receptors can exist in different states.
In different states. And that's super important for regulating how they respond to light.
So just like those.
Yeah.
Inhibitory interneurons.
Yeah.
In the spinal cord.
Yeah.
They're using glycine.
They're using glycine to fine-tune muscle activity.
Yeah, something similar.
Something very similar.
Must be happening in our eyes.
Yeah.
To regulate how we respond to light.
You got it.
Okay, I'm starting to see where this is going.
All right.
This is really cool.
So let's finally bring this back.
Okay.
To those photoreceptors.
Yes. Rods and cones.
Rods and cones.
Right.
And the phototransduction cascade.
That magical cascade.
That magical cascade. So remember, rods are for dim light.
Dim light.
Cones are for color vision.
Color vision, okay.
And both of them.
Both rods and cones.
Contain these light-sensitive pigments.
Pigments?
Yeah.
Called opsins.
Opsins, okay. They sound important.
They are key to this whole process.
So the opsins are like...
Yeah, they're like little antennas.
Little antennas that capture the light.
They capture the light, exactly.
Okay, I'm with you.
And in rods, the specific auxin is called rhodopsin.
Rhodopsin.
Yeah, have you heard of that one?
I have heard of rhodopsin. I feel like that's a big player in the world of vision. So
when a photon of light hits a rod cell, it gets absorbed.
It's absorbed by rhodopsin.
By rhodopsin, and that triggers
a
whole cascade of molecular events.
Okay, so it's like
a chain reaction.
A chain reaction.
Yeah, and every step amplifies the signal.
So it starts small.
It starts small and gets bigger.
And just amplifies
as it goes.
Yeah, like a domino effect.
Okay, so rhodopsin gets activated.
Rhodopsin gets activated.
By light.
Yeah.
And that sets off the cascade.
It kicks it all off.
But what's actually happening in this cascade?
Right, so...
How does that ultimately lead to us seeing?
So this cascade ultimately leads to changes.
Changes. What kind of changes?
In the concentration
of what?
Like of a molecule called cyclic GMP.
Cyclic GMP.
Yeah, or we can call it CGMP.
CGMP.
It's GMP inside the rod cell.
Okay, wait. CGMP.
Yeah.
We were talking about...
You're talking about...
Cyclic AMP.
Cyclic AMP. Are they related?
Are they similar?
They are similar in that they're both signaling molecules.
Okay. They're both messengers.
They're messengers, yeah. Involved in all sorts of different cellular processes.
Okay, but in the case
of
phototransduction,
yeah.
CGMP has a specific role.
Okay, what does it do?
It regulates the flow of ions.
Okay. Ions are charged particles, right? So it's regulating
across the cell membrane.
The flow of charged particles.
Yeah. The rod cell. Okay, I'm listening.
So in the dark,
okay.
CGMP levels are high.
High CGMP in the dark.
Inside the rod cell.
Okay. And so what does that high CGMP do?
That high CGMP keeps certain ion channels open.
Ion channels open.
Yeah.
Okay. What's flowing through those channels?
Sodium ions.
Sodium ions.
Are flowing into the cell.
Okay.
And this inward flow of sodium.
Okay. Sodium's positive, right?
Sodium's positive, yeah. Keeps the rod cell in a slightly depolarized state.
Depolarized?
Yeah. Remember depolarization.
Remind me, what does that mean?
It's like a car.
Okay. I like this analogy.
With its engine idling, ready to go. It has a slight...
It's ready to fire.
Positive charge inside compared to the outside.
Gotcha.
So in the dark.
So in the dark, the rod cell is idling.
The rod cell is idling thanks to that high CGMP.
Thanks to all that CGMP. But what happens when light comes in? What happens to the CGMP?
When light hits rhodopsin, it sets off...
It starts the cascade.
That whole cascade we were talking about.
And what's one of the key players?
One of the key events
in this cascade
is the activation...
The activation
of an enzyme called phosphodiesterase.
Phosphodiesterase.
Yeah.
It's a mouthful.
That's a long word. But it sounds important.
It's a very important enzyme.
Okay, what does it do?
So, phosphodiesterase is like a CGMP chomping machine.
A CGMP chomping machine.
It breaks down CGMP.
Okay, so it gets rid of it.
It gets rid of it, and it causes those CGMP levels inside the rod cell to plummet.
To go down.
Just drop way down.
The light comes in.
Light comes in.
Yeah.
Activates rhodopsin.
Activates rhodopsin.
Triggers the cascade.
Triggers the cascade. Activates phosphodiesterase.
Activates phosphodiesterase.
And that phosphodiesterase, CGMP levels drop.
Gobbles up all that CGMP.
What happens to the rod cell?
Okay, so remember those ion channels?
The sodium channel. Yeah, the sodium channels that were open because of all that CGMP.
Okay.
When CGMP levels drop.
When the CGMP drops, those channels close.
Those channels slam shut. And they prevent...
They're like, no more sodium.
No more sodium allowed. So that influx of sodium ions stops.
Okay.
And without that, continuous flow of positive sodium ions, the rod cell becomes hyperpolarized.
Hyperpolarized.
Yeah. So it's the opposite.
So it's the opposite of depolarized.
Of depolarized. So instead of the car idling...
it's like...
It's turned off.
We've turned the engine off.
Okay.
It shifts to a more negative charge.
More negative.
Inside. Compared to the outside.
Gotcha. So it's all about...
In this change in electrical charge. That's the signal.
That's the signal.
Yeah. That gets sent...
That says, hey.
Down the line.
To your brain.
Say, hey.
I saw that.
I detected light.
Okay.
So to recap, light comes in.
Light comes in.
Hits rhodopsin.
Triggers the cascade. Triggers the cascade. Activates phosphodiesterase.
Activates phosphodiesterase. CGOP levels drop.
CGMP levels plummet.
Yeah.
Ion channels close.
Ion channels close. Rod cell hyperpolarizes.
Rod cell hyperpolarizes.
We see.
We see.
That's incredible.
Isn't that amazing?
And all that can be triggered
by a single photon.
Can be triggered by a single photon of light. Talk about sensitivity.
That is amazing. Our eyes are so sensitive.
It's mind-blowing.
We're not done yet, right?
We're not at the finish line.
Because that signal still has to get.
It still has to get.
Yeah.
From the eye to the brain.
From the eye to the brain. So how does that happen?
All right, so the hyperpolarization of the rod cell triggers a chain reaction.
Another chain reaction, okay.
It involves other cells in the retina.
Other cells. So it's not just the rod.
It's a whole team effort.
And cones. Okay, so who are these other cells?
And ultimately, it leads to the activation of ganglion cells.
Ganglion cells.
What are those?
So ganglion cells are the final.
The final output neurons?
Output neurons of the retina.
Output neurons of the retina. Okay, they have these long axons.
Long axons?
Yeah, those are like cable-like projections.
Okay.
And those axons form the optic nerve.
The optic nerve. That's the highway.
That's the information highway.
To the brain.
From your eye to your brain.
Okay, so the signal from the rod cell.
So the signal, after it's been processed.
It gets processed by all those other cells.
By the other cells in the retina. And then
passed on.
It gets passed on.
To the ganglion cells.
To the ganglion cells.
From the ganglion cells. transmitted
via the optic nerve.
via the optic nerve to the brain.
The brain.
It's like a relay race.
It's like a relay race?
It's like a relay race.
Yeah. The rod cell passes the baton to the other cells
in the retina.
And then finally, the ganglion cells
The ganglion cell
carry that signal all the way to the brain.
all the way to the brain, to the finish line.
It's a beautiful thing. I love it.
It is pretty remarkable.
That's incredible. It's like this whole relay race happening in our eyes.
It really is. And it all
Yeah.
happens so fast.
Yeah, it's amazing how something so small, like a photon,
Right. A tiny little particle of light.
can trigger this whole chain reaction.
It's mind-blowing. And it all leads to us being able to see the world.
Yeah, it's like this whole symphony of signals.
Totally. A symphony of vision.
No, we've only talked about rods so far, right?
Yeah, we've been focusing on those.
Those are the dim lights.
Dim light specialists.
Yeah.
But what about color?
Right. How do we see all
How do we see those vibrant colors?
the beautiful hues of the world?
Yeah, that's where the cones come in.
Exactly. The cones are all about color.
So cones are like rods.
No way. Yeah. They both have opsins.
They both have opsin.
Cones have a different kind.
Okay, different kind of opsin.
Yeah, one that's sensitive to different wavelengths of light. Different wavelengths of light. Okay, I'm listening.
So instead of rhodopsin,
Okay, so no rhodopsin in cones.
cones have their own special opsins.
Okay, so there are multiple types of opsins.
Yeah, we actually have 3 types of cones.
There are 3 types.
Red cones,
Okay.
green cones, and blue cones.
Red, green, and blue,
Yeah.
like the primary colors of light.
Exactly. And each type of cone
Okay.
contains a slightly different opsin.
Okay, I'm with you.
So the red cones
Yeah.
are most sensitive
Okay.
to long wavelengths of light.
Long wavelengths.
Green cones are sensitive to medium wavelengths.
Medium wavelengths.
And blue cones are sensitive to short wavelengths.
wavelengths.
Okay, so you can think of it,
So each cone is like
each cone is tuned.
tuned to a specific color of the rainbow.
Yeah, like a little radio
Do it.
tuned to a specific frequency.
Okay. So then how do
And then our brain takes all those signals
Huh.
from those 3 cone types and combines them
Yeah.
to create the
Yeah.
perception
Of all the colors.
of a full spectrum of color.
That's amazing.
Right. So your brain is like,
It's like an artist.
yeah, a master artist.
That's mixing the reds and greens and blues
Blending those signals to paint a picture.
to paint the picture
Yeah.
of color vision.
No, we see.
Pretty.
That's so cool.
It's pretty remarkable.
Okay. So we've got
You got that.
rods for dim light, cones
For color.
for color.
They both use opsins.
They both use opsins.
They capture light. They triggered that phototransduction
That
cascade.
amazing cascade
Yeah.
leading to
Those signals get sent to the brain.
through the optic nerve.
It's amazing how it all comes together.
It's beautiful, isn't it?
It really is.
This whole system.
Well, listeners, I don't know about you,
Yeah.
but I am feeling a whole new level
Yeah.
of appreciation for my eyes.
I know, me too.
We started with this puzzle
Right. Totally unrelated.
about the heart.
Yeah.
We learned about
We took a detour.
microspinal cord and AKPs. We took a detour
We learned about glycine.
to the spinal cord
Yeah.
to learn about glycine.
Yeah.
And we ended up in this incredible world.
And it all ties together.
This division.
Yeah. It just goes to show.
And it all came from these seemingly unrelated sources.
Sometimes the most unexpected connections
Yeah.
can lead us to the most profound understanding.
That's the beauty of science.
It is.
So we'll leave you with this.
Sure.
If hormones, like thyroid hormone,
Yeah. Abitar.
can influence the development of our photoreceptors,
Mhm.
what other factors in our body
What other thing
might be shaping how we see the world?
could be influencing?
Could it be the food we eat,
Yeah.
the environment we live in, the experiences we have?
The people we interact with, it really makes you think.
It really does. It makes you wonder how much of what we see is influenced by
Yeah.
factors outside of our eyes.
How much is our biology? And how much is everything else?
It's a fascinating question,
It really is.
and one that I think
Yeah.
deserves further exploration.
Absolutely.
Until next time, keep exploring,
Keep those eyes open.
keep questioning, and keep those eyes wide open
Yeah.
to the wonders of the world.
To wonders of vision.